doctor, every hospital, every nurse have been notified. Every woman in this country, I think, must be aware that it's most important that they check their medicine cabinet and that they do not take this drug, that they turn it in. Every citizen, of course, should be aware of the hazards.
Back in November of 1956, a West German pharmaceutical company by the name of Chemie Grünenthal developed a product that not only treated colds, the flu, and nausea, it was deemed to be incredibly effective against morning sickness in pregnant people, therefore a much-needed lifeline in an otherwise stressful period in their lives. In comes thalidomide. Early testing from their company researchers found that this wondrous compound had an incredibly high LD50 value, which meant that even at large doses, thalidomide was considered to be a relatively harmless compound with very little downside. It also appeared to be exceptionally well tolerated, was a compound that was not habit-forming, and just generally very safe, with prolonged sleep being one of the side effects of taking the drug. With such a beneficial effect and virtually no issues at any level of dosage, thalidomide was licensed in July of 1956 for over-the-counter sales in Germany. Eventually, other companies caught wind of the incredible benefits of thalidomide. Around the world, and under a license from Chemie Grünenthal, a total of 14 pharmaceutical companies started to produce thalidomide in 47 countries across 37 different trade names. Distaval and Asmaval were some of the thalidomide varieties that spawned from the success in Germany, with advertisements that claimed, Distaval can be given with complete safety to pregnant women and nursing mothers without any adverse effects on the mother or the child. For a few months, this drug was living up to its reputation as a safe and effective method of dealing with morning sickness in female patients, and with a standard toxicity tests that Chemie Grünenthal was performing in mice, there was really no reason to expect a negative outcome. In 1960, a general practitioner from Turriff, Scotland, by the name of Dr. Alexander Leslie Florence, was the first healthcare professional to publicly question these safety assumptions in a letter that he wrote to the British Medical Journal. He reported that in 4 of his patients who had taken thalidomide, there was development of severe paresthesia, which is a pins and needles numbing sensation in their hands and feet. These people also experienced coldness in their extremities, along with nocturnal cramps and occasional slight ataxia, which is the loss of coordination within the muscles. He also provided the journal with physiological observations when the compound was removed, stating that 3 of the patients now received no thalidomide for 2 to 3 months, and there has been a marked improvement in their systems, but they're still present. It would appear that these symptoms could possibly be a toxic effect of thalidomide. And so, even with these initial side effects at the time, concerns about the drug weren't high enough to warrant it being pulled from the shelves, and it wasn't until a notable individual started to take a closer look. And that's where we meet Dr. Francis Kelsey.
A stubborn stickler of a medical officer in the Food and Drug Administration, Dr. Francis Kelsey, who came originally from Cobble Hill, British Columbia, and was educated at McGill University in Montreal.
Dr. Kelsey, who was new to the FDA in 1960, was assigned to investigate a thalidomide product, Kevidon, which was produced by William S. Merrill Lab, located in Kansas City, Missouri. To Dr. Kelsey, a compound like this seemed to be an anomaly, stating that it was just too positive and that this couldn't be the perfect drug with no risk. At the time, during Dr. Kelsey's FDA investigations, Kevidon was being aggressively campaigned for approval, as the people in charge were enamored with the numbers coming out from West Germany. Over 1 million doses a day of thalidomide were consumed back in 1960, and people in America wondered if that sort of success could be replicated on home soil, which also happened to be the largest and most lucrative drug market on the planet. Expectations of blockbuster profit margins pushed their lobbying attempts to the max, which caused Dr. F. Joseph Murray, who was a Merrill executive then, to immediately approve the sale of Kevidon in the United States. Dr. Francis Kelsey had something to say about this insistence, and asked for hard evidence from Merrill to back up their drug's safety. The initial Merrill application was heavily reliant on anecdotal testimonies in place of bioassay calculations, and because of this rather unusual proportion, she even referred to her NIH-employed pharmacologist husband, who hilariously stated that the application was an interesting collection of meaningless pseudoscientific jargon apparently intended to impress chemically unsophisticated readers, along with the very unusual claim that thalidomide had no LD50. Dr. Kelsey's already high level of concern was further elevated when she came across Dr. Alexander Leslie Florence's medical observations on thalidomide in the British Medical Journal, specifically concerned with the incidence of peripheral neuritis, which is nerve damage in the extremities of the body like hands and feet. Given such an alarming and direct connection to a widespread compound and a rather disturbing physiological side effect, Dr. Kelsey told Murray that the burden of proof that the drug is safe lies with the applicant, and that in this connection, we're much more concerned that apparently evidence of peripheral neuritis in England was known to you, but not forthrightly disclosed. Well, shit. This was bad news to the people behind Kevidon, including Dr. Murray. He tried to reason with Dr. Kelsey's boss in an attempt to call the observations as somewhat libelous, and if there was any way that the letter was subject to reconsideration. And spoiler alert, it wasn't. And in fact, the peripheral neuritis report prompted Dr. Kelsey to request further proof that the drug was not harmful to the fetus inside of expecting mothers. Dr. Kelsey had rejected the insistence 4 times to reject Merrill's application on the grounds of insufficient data, which was great because, at the time, reports in Europe and Australia had started to come out stating that spikes in birth defects were appearing and thalidomide was the common denominator. Turns out that this thalidomide drug is a teratogen, which are substances that have the potential to cause non-heritable birth defects via a toxic effect during early development. Because thalidomide was able to pass through the placental barrier of expecting mothers and directly interfere with fetal development, it was not only able to apply teratogenic effects on the formation and function of the placenta itself, but also affect nutrient absorption and general growth of the fetus. But why is that? Preliminary tests were done on mice, which was the standard protocol at the time, so how was such a widespread disaster like this possible if a test was done to screen for possible side effects? Back in the 1950s, Gruenthal's research team were unaware of a critical factor in distinguishing teratogenic markers since their initial tests were only done on mice. Turns out that because thalidomide doesn't increase the amount of certain proteins in their embryo cells, they're able to prevent the induction of apoptosis, or programmed cell death. More modern research has shown us that in other organisms like rabbits, chickens, and humans, there's no prevention of induced apoptosis, so while we'd be considered to be thalidomide sensitive, mice would be thalidomide insensitive. This susceptibility originates from a single amino acid difference in the cereblon protein. Primate varieties and rabbits have a susceptibility for thalidomide to have teratogenic effects on the embryo, whereas the presence of an isoleucine at the same position predicts a lack of thalidomide teratogenicity. Mutant mice with a valine in this position in question, compared to wild-type mice which sported isoleucine, showcase statistically more miscarriages than their unmutated wild-type mice. Thalidomide induces increased fetal demise of mutated cereblon litters, but not wild-type litters, with data showing a loss in pregnancy and increase in fetal complications in the mice that have the valine amino acid compared to those with isoleucine. And as you would imagine, the fallout from this miracle drug was absolutely immense. Several countries reported thalidomide-related defects and deaths that exceeded 1,000 individuals, which was then followed by Cathy Gruenthal, Distillers Company, and a variety of other companies that manufactured thalidomide going to trial in the 1960s. Financial compensation settlements were reached, totaling over $100 million across multiple countries for the victims that were affected by the drug and still alive but suffering from the ongoing effects. And oddly enough, there were some positive things to note despite the absolute devastation of the crisis. The first was recognizing Dr. Frances Kelsey and her work. Her insistence on the FDA's refusal to approve thalidomide's sale in the US reduced the number of potential cases by a significant margin. The compound still made its way to doctors and patients through unapproved means, but with the perceived efficacy and safety of it at the time, it wouldn't shock me to see estimates of 100x or even 1000x without Dr. Kelsey's constant intervention. She did receive the President's Award for Distinguished Federal Civilian Service from JFK directly in 1962 for her absolutely critical contributions during this crisis. Also in the US, the thalidomide crisis directly led to legal reforms. On October 2nd of 1962, the Houses of Congress unanimously passed new amendments, which then President JFK signed into law just 8 days later.
Every woman in this country, I think, must be aware that it's most important that they check their medicine cabinet and that they do not take this drug, that they turn it in. Every citizen, of course, should be aware of the hazards.
This basically means that they now require manufacturers to provide both data from the animal experiments and highly standardized trials in humans, proving that a drug is actually safe and effective before putting it out in the market. There were also a variety of other improved FDA amendments that went into effect to ensure public safety, along with improved laws worldwide, like the 1968 Medicines Act, which drastically improved the distinction between medicine and prescription-only medicines. They brought this idea into the mainstream public that perhaps drugs weren't this thing that had no downsides, and maybe we should take precautions in distinguishing what is actually safe versus what needs a little bit more control. Dr. Kelsey then continued to work for the FDA until her retirement at the age of 90 back in 2005. She then had a school named after her, was named into the National Women's Hall of Fame, and even received the Order of Canada, which recognizes extraordinary contributions, even though most of her work was done in the US. She unfortunately passed away in 2015, within 24 hours of receiving the insignia for the award, but not without millions of people around the world thanking her for her heroic efforts across multiple decades.